have all the relevant data that is required. For example, your toxicity in animals, the safety in animals, the efficacy, and all the other things. Okay, so in order for you to get all of that sorted, once all of that has been done, you will have to go to um, you would then have to go to your clinical trials. Okay, so normally after your animal studies here with the animal models, with your in vivo studies, this is when you move on to the next step. Now, before I move on to that part where you all present, I would also like to share with you all this clinical trials. Give me a second. Clinicaltrials.gov. Okay, let me share this page. Clinicaltrials.gov. So I hope you all were able to have a look at this website, right? Because most of you would have gone through whatever that you need, your examples and everything from this website. Now, the good thing about this website is that access is actually free. Why? Because all the data that you've done for any clinical trials must actually be inserted inside here. Okay, so when you do a search with clinicaltrials.gov, there are a few things that you can filter out. Okay, so you can filter by condition, by whatever terms, whether your intervention is it an intervention or a treatment, or maybe even diagnostics, and then you can even go by location. So let's try, for example, yeah, this one says heart attack. Okay, let's just go by heart attack. So when you look at heart attack, so what you normally get is that you will normally find all your search result that actually includes heart attack. Okay, so the thing that you have to be aware when you look at clinical trials, clinical trial website, is that there are a few things that are important. So the things that are important is that you will see this is the file number for the clinical trial. Okay, so this title is normally the title that is given by the PI or the company. Okay, and then what is the condition? Okay, now although we put heart attack, all these other conditions, if you look at this is myocardial infarction, this is periprocedural myocardial necrosis, and etc. So all of this is basically conditions or keywords that is used for you to find out what is it that you want to know. Okay, so once you have done these basic ones, you will then see what are the other terms. So what other terms, for example, you have heart attack and say, for example, vaccine injury, for example, the one that's caused by the mRNA vaccine. Okay, and then you want to see the intervention. So the intervention could be the name of a drug, okay, or it could be a name of a method, or if it's for diagnostics, sometimes you can also include that. Okay, and then location, no need to say. But what is also crucial is that you have a look at this. You see on this top part is either written as completed, unknown status. There is also active. Let me see where I can find. Okay, there are some which is terminated. Okay, and then let's see if there is some that is written as active. This is withdrawn. So terminated means most likely it has there might be some issue with it, so they terminated it. Withdrawn is most likely they don't have enough patients to take in. Okay, so when they don't have enough patients, sometimes they just withdraw the deal. Okay, and then there are some. Let's see what are the other examples that's available. Yeah, okay, look at this. This is recruiting. Okay, so recruiting here means that they are still finding more patients to participate. Okay, and then this is not yet recruiting, which means this is relatively new. So this one is in Jindan, Shandong, China. Okay, and then terminated, unknown status. Okay, look at this. This is active and not recruiting. All right. So this one here means that the clinical trial is ongoing, but they don't recruit anymore. There's a few reasons why they don't recruit anymore. Most likely it's because they have achieved the number of patients that they want. So when they have sufficient number of patients that they want, they'll just put a term there, I'm not recruiting anymore. All right. So after this, when you click into each and every one of the clinical trial, you will be able to find more details on clinical trial. Okay. So study number, this is the NT number that you can see from the top. Okay. Then you can also see some of them, they will have sponsors. Okay, the sponsors means that it's funded by who. Like this one is sponsored by the university. Sometimes some of these could be sponsored by big pharma companies, and then all of these can then be captured. Okay, so then the person who provides the information is this person, Michelle Dorset. Okay, so he's the responsible party, most likely the principal investigator. And then you also have last update posted. Because the thing is that all this information that is updated, the website doesn't go and ask for an update. Okay. So the person who is responsible, the PI here, will have to update their findings here, especially if they want to um, move their things from stage A to stage B of the clinical trial. They need to update it regularly. Why? If they don't upgrade this regularly in the future, if they want to apply for FDA, all of that will be a problem. Okay? So then here you will be able to see the study overview, location. Okay, this is something which is crucial. Whenever a clinical trial is done, there is normally criteria. Okay, so it's called the eligibility criteria here, and normally it includes two. One is called the inclusion criteria, the other one is called the exclusion criteria. Okay, inclusion criteria means they want to target specific population of people. Okay, so for example, this one, they say, oh, they are willing and able to access the internet to participate with the SMART program, score of PPS 10 or 14 or greater. So this one, I think, is heart disease. Yeah, so this one might be stress management or something like that. And then concurrently enroll in an observational study of patients who had acute myocardial infarction. So the thing which is excluded is here the person is poorly controlled physical or mental health condition, will interfere with the ability to participate in SMART, and then current daily practice of meditation of 10 minutes or more per day. So these are all the criteria that is excluded. Okay, so sometimes there are some, for example, if it's a stage one or phase one clinical trials, especially for healthy patients, the exclusion criteria could also be things like pregnant mothers. It could be somebody who has certain illnesses. So all of those will be included in the exclusion criteria. Okay, so this is inclusion, exclusion. And then in terms of ages, normally they will also define. Now, why do you think that they will put 18 years old and older normally? Anybody got any idea why? Why do you think they would be normally stating a certain age or older? Anybody? Or everybody has this problem?
Maybe only 18 years old, yeah, you can responsible for yourself. You can sign the thing.
Yes, so that is the key thing. Whenever somebody does a clinical trial, the person would have to give consent. Okay,
Yeah,
so consent is very important. So normally they want somebody who is 18 years old and above because these are the people who can consent. However, if for example they are clinical trials that is specific for children, say for example, what sort of example? Um, Maybe patients with hole in the heart. When they are born with hole in the heart, or patients who have certain disabilities, or even children who are born with some children are born with leukemia or whatever. So those are things they might also put age eligibility, but maybe they will say okay 2 years old and above, or between 2 to 5 years old, but consent must come from parents. Okay, so you're right there whereby consent is very critical. Okay, so then after you see the participation criteria, which includes all the eligibility, then they will also then show you the study plan. Okay, so for the study plan, they will say, okay, what is the purpose of the work? What is the intervention? Masking here doesn't mean masking. It's just basically masking of certain data. Or for example, if let's say a medication is given, whether the data is being, whether the medication is given as a placebo or whether there is placebo involved or not. So these are the masking, okay? And then what are the arms? So what they do, these are all the details. And then the other thing is that whenever you do clinical trials, measurement must be important, which means that all the things that you state must be measurable. Example, blood pressure, certain cytokine level, or oxygen level. So all of this must be measurable. So like this one, change in amygdala activation. So you can see they put a radio tracer or whatever, and then they have a time frame for them to do. Okay. So after that, you have collaborators and investigators. So if let's say there's only one PI, then they'll just put one PI. But if there are multi-place collaborator, then they'll just put a collaborator there. Okay. And then publication, this one is basically the information. If let's say they publish data, it will be put in. But for publication, normally is when the study have ended and has yielded a certain positive result. Okay, negative results also they will publish but it's just that sometimes most people will want to publish when it's positive. Okay, and then a lot of companies, pharmaceutical companies, they won't put publication, but be it positive or negative. So the only time they will put in the publication is when, for example, it is put in a certain manner or in a certain institute, and then the institute want to publish the data and they agree to it, then they'll publish. But normally for pharma companies, they don't want to publish it. They just put all the data inside here, and then once it and then it will be submitted to the regulatory department, FDA and all those things, and then only they will proceed. Okay. Then you have the final bits, which is all the record history, the information on the gland of the participants' data, and then what are the name of the drugs and everything. Okay. So this is how you basically look or use clinicaltrials.gov. So for you guys now, maybe you feel that, this is not really my problem, but it is very crucial, especially when you are at a level where you want to design your own therapeutics or diagnostics, which you hope to one day be put into human. You should actually have a look at this because it basically gives you an idea of how you need to plan your work from the beginning. Okay? Because if you are able to plan your work from the beginning, all of the things will be much more smooth sailing. It won't come to a point whereby, wow, your thing is working, but when you try to apply for a clinical trial, then you realize there are some holes in your current data, or there are certain things that you haven't done, or certain things that you haven't thought of. Okay. So, for example, if let's say you want to do gene therapy for a certain disease, so you might want to look at trials.gov to see if there's any gene therapy that has already been done. And that basically gives you an idea of what you actually need to put into in order for you to achieve whatever you want to achieve in the future. Okay. So this one is researcher's view. Okay, this is all the information, pretty much the same, but it's much more concise. Okay. And then, okay, this one written there, there's no result posted because this is still active, but it's not recruiting because it's still running at the moment. So they hope to complete, this is estimated only a completion date by December 2023. So maybe the thing has completed, but they haven't finalized the data to be published. So they haven't put anything here. And then last, your record history. So this is the same thing, whereby when they update certain things, and the last updated was this date, and at this date is basically the status of the recruitment, the study status, and the study design. Okay, so this is basically how you look at clinicaltrials.gov. All this information is critical, especially for you guys who want to hopefully one day be in a position whereby you have a certain discovery that you made, and you hope that that discovery can go into proper use for humans. Okay, so if you study and you understand the clinical trials part, you would then be able to have a much more clear indication of what you need in the future. Okay, can understand people? Can Okay, so that's all from me today. Now, we're going to start Any question before we start our presentation? Any question?
No.
No question See lah, online online like that lah, cannot see anybody reaction.
Yeah, you want to see my face, is it?
No lah, I don't want to see your face. I just need to see reaction so that I know whether you understand or not. If you look
Okay lah,
blurred,
okay
I
lah.
know you don't
Okay lah,
understand.
okay.
Okay, so let's move on. You kahwin lah.
This one last time one.
Okay, now let's start with the first group. So Yi-En and Sana will be presenting on the phase 1 clinical trials. So Yi-En and Sana, you guys go ahead. Now, the thing is that once you're done with your presentation, I want you all to submit your presentation into the link that I've given. All right. There will be no grade involved. It's just that I want you all to put that in so that everybody else will be able to download other people's one to read. Okay. So it becomes like you're preparing notes for yourself, but at least you don't prepare it by yourself. You're preparing it with your friends. Okay. All right. So Ian and Sana, you all can go ahead.
Yes, we are ready.
Okay, go ahead.
Can you see my slide?
Yep, I can see it clearly.
All right. Good afternoon, everyone. So today, me and Sana will be presenting about clinical trial phase 1. So as Dr. Tai has already told us that drug discovery until the phase that it's marketed and used in clinical settings takes a very, very long time. So this drug will go through many phases before it is marketed in the clinical settings. And let's go into some details before the clinical phase 1. So preclinical phase usually is investigating the pharmacodynamic studies in which it investigates the effects of drugs on body system, while pharmacokinetic data was also obtained during the preclinical phase where they investigate the effects of body system on the drugs. And here they also calculate the dose range and study the toxicokinetic studies of the drug before it is used on human. So this is usually done on animals. So the major thing that I want to emphasize on preclinical phase is that they are used on cells or animals, but not humans. So what is the difference of preclinical phase and phase 1 clinical trials is that phase 1 clinical trials is done on human body system. And the main objective of this phase 1 clinical trials is to determine the safety, tolerability, pharmacokinetic, and pharmacodynamic of this drug on human. So they usually use healthy individuals or patients with specific disease, depends on what drug that you are testing for phase 1 clinical trial. If you are looking at the FDA definition of phase 1 clinical trial, they usually have participants of 20 to 100 healthy volunteers or people with specific disease with the length of study is several months. And the purpose, again, it is very important to determine the safety and dosage of the drug. And most of the drugs will move on to the next phase, which is phase 2 clinical trial. If you are looking at Malaysia guidelines, okay, there are typical number of subjects that are used according to Malaysia guideline. Clinical trial phase 1 is 50 to 200 subjects, which the subjects are healthy or specific with patients with specific disease and are not expected to benefit from the drug investigational product. Okay, so the objective again is very important. This is the third time that I'm saying the objective of the clinical trial is to study the PK, PD, and the safety of the drug. Okay, so phase 1 clinical trial is a very exploratory phase as this is the first in human clinical trial because this drug is previously always used on cells and used on animals but not been used on human before. So it is a very exploratory phase on human And at this stage, only preclinical data are available to guide the dose selection, population, study design, safety monitoring. So an appropriate expertise and are very important for us to maximize the safety of the study subjects and quality of the data. So these new drugs or new compound that we are testing are usually tested on healthy volunteers with increasing frequency and ascending dosage. Next slide. Okay, so there are few key aspects in first in human study in which there include the ones as shown here: choice of study population, study design consideration, etc., etc. So let's go one by one into it. So the choice of population in phase 1 clinical trials, most of the studies, they will rather use healthy subjects, as when you use healthy subject, there are many advantages, including the speed of recruitment, because when you are recruiting patients with specific disease, you might take longer time to recruit them, and the concomitant medication and disease pathology in patients can be eliminated, because when you use healthy individuals, they are not taking any medication. Hence, there is no medication that will interfere with your current clinical trial. And healthy subjects may generally tolerate more intensive intervention and adverse effects as compared to that of the patients. However, some of the studies still use patients as their main subjects, especially oncological agents, where they test on a cancer patient. And most of the study, they use bridging study design, as they use healthy subjects for lower dosage to facilitate the improved dose selection. And lastly, they will use a patient with a regime of higher dosage. So this will give a more informative safety data and pharmacodynamic data with the clinical trials being done. Okay. So these are the pros and cons of when you are using healthy subjects and patients, and why you are using them. Yeah, so this is just for your reference. Next slide. How to go? Okay. So next is the study design consideration. When you are doing phase 1 clinical trials, which is a first in human trial, it is important to consider the compound characteristic and timeline and site logistic. So there are two major study design that you can use. The first one is sequential group design, and then the second one is a crossover study design. So sequential group design usually uses single ascending dose escalation in healthy subjects. And this kind of study design usually used when there is a concern to expose subjects to more than one dose of active drug, or biologics when neutralizing antibodies could be formed, or when as simple as when the sponsor just want more subjects to look at the subject variability. However, the disadvantage of using this study design is that you have to recruit a lot of people. It means like 3 times of the number of subjects required for crossover design. So it depends on how you want to design your study and how many people you are recruiting and what are the drugs that you are testing. Next is the crossover study design. So crossover study design are also split into two main categories, which are sequential and alternating crossover study design. Okay, so sequential is usually you have a dose that is escalated within a cohort. So here you can see that you only recruit 2 cohorts of patients as subjects, as compared to the previous study design that you recruit 6 cohorts of patients. So this will reduce the number of subjects that you are recruiting. And then usually within subject, dose increments will be small when you give the drugs, and wider dose range can be studied with this kind of sequential crossover study design. And then lastly is the crossover study design, which is the interlocking or alternating cohorts, in which you give this drug to cohort 1 and then increase the dose in cohort 2 and increase and some more in cohort 1 again, and so on and so forth. So this this study design is usually used for drugs with longer half-life, as you want to increase the washout periods between the subjects and reduce the risk of pharmacodynamic and pharmacokinetic carryover. So those increments in this study are often larger, and you need longer time for this kind of study. So the disadvantage is that you may increase subjects dropout. So after you have got your study design, you have to determine what is the starting dose for first in human, right? Because previously you are always testing on animals. So what is the starting dose on human? So previous guidance recommend that the starting dose should be not more than 10% of predicted therapeutic dose. And if you are looking at FDA guidance, they have recommended the stepwise process as shown here. They have to convert the no observed adverse effect level from toxicological study to a human equivalent dose based on the body surface area. And then they select the HED from the most appropriate species, apply safety factor of more than or equal to 10-fold to give a maximum recommended starting dose. And then they adjust this based on the previous clinical study pharmacological action of the drugs. Okay. So after you have determined what is your starting dose, when you are doing the clinical trial, you have also to increase the dose levels by levels. So the ascending dose trial is usually increased in 3 to 5-fold of increment when the drug is at lower dosage. And when you increase until the drug is around the therapeutic range, the smaller increment should be expected. And the general rule of this dose increment is that dose to toxicity or dose effect relationship observed in the non-clinical studies have to be considered. And depending on which one is steeper, dose and toxicity or dose to effect relationship curves, the lower the dose increment should be selected. Next is that when you recruit your subjects, it must be based on the subject's free will, means you cannot force them, okay, come to my study because something, okay? The recruitment should give a valid consent and fully and properly inform the subjects on the following things, which are what is the study and what is the purpose of your trial, and what are the risks that are involved in this trial, and they have to know that they are fully on their free will, means that they can withdraw at any time and without giving any reasons, and the principal investigator can terminate them if they are not following the protocol of the clinical trial. So, but there are some compulsory things that you have to look at when you are recruiting subjects, which subjects must not take part in more than one clinical trial, so that these two clinical trials will not interfere with each other. And the subjects must not receive more than 10 microSV of radioactivity in any 12-month period of a healthy subject. Means if the subject was previously participated in clinical trials which also use radioactive drugs, and now you are the healthy individual want to participate in some more clinical trials that use a radioactive drug, and it exceeds the level of this 10 microSV, then the subject cannot join this clinical trial. And lastly is the informed consent. So informed consent of the clinical trial phase 1, it requires oral and written information which are free of jargon, and they have to give the subjects the opportunity to ask questions about the trial, and the subjects cannot be forced by the investigator or the staff to join or continue to join this trial. And all the new information that was obtained in this clinical trial must be given to these subjects. And lastly, the subjects have to sign and give the date on the consent form and are given a copy of this consent form. So after the first human clinical trial, what is next? So usually the principal investigator will also evaluate a lot of data, including to further contribute to the characteristic of this drug, which include the food, the potential influences such as food, gender, age, genetic, ethnicity difference on the pharmacokinetic of this investigational drug. And they will also evaluate some interactions such as drug-drug interaction, ADME of radio-labeled drug if you're for your drug, and then you also check on the bioavailability and bioequivalence of this drug. And in some of the studies will also include the electrical activity in the heart, which measured the QT interval of the electrocardiogram. And that's all for my part. So this one will be Sena's.
Go ahead, continue, Sana. Everything okay, Sana?
Sorry, I don't know what happened with my. Okay.
You must have opened too many things,
Just
and
I
your
was.
application might have hanged. Just
Okay.
a gentle reminder to everybody, WebEx takes a lot of resources, so you might want to close most of your
I don't know what's.
application before you start
I
sharing.
don't know what's going on with my.
Sana, do you want me to share the slide? Then you just tell me when is it the next slide?
Yes, please.
Sana, can you see the slide?
Yes, thank you so much. So, can you hear me?
Yes, loud and clear.
Okay, sorry for that mistake. As like Yin, as our part like with Yin, we have we had a presentation about clinical trial in phase 1. So in the first slide, like I want to share about the most clinical trials that initiated in the 2023. It was phase 2, and our topic is Yin. Can you just like move the like just the slide? So the clinical phase in the the number of the study in the phase 1, it was like 22% 22 to 23% of the whole clinical trial that have been started in the 2023. And as a number, like nearly about 40,000 of study have been started and in active state, and also the completed one. Yes, Ian, just continue. Okay. On the world map, clinical trials in phase 1, it's conducted on the worldwide. Most of the clinical trial in phase 1, it's conducted in the USA. It was about 20,000. And my points here, it's about all the clinical trials. Like you can just go, like, please. Again. Can you just help with the, okay, just passing the slide because there's a lot of the movement with that thing. Okay, all the cases like here's in the phase 1, they are completed or in active state. And then I focused on the Malaysia numbers. So in the Malaysia, we have only 36 clinical trial in phase 1 that have been completed totally. So, but mostly of the study clinical trial in phase 1 in Malaysia, like conducted in the hospitals, governmental hospital, and also the university things. And here's like pointed out, like all clinical trial in phase 1 mostly conducted in between China's university with the Malaysian university. So it is just kind of the positivity, like a collaboration between two countries about done in the clinical trials. So next slide. And this is all the list of the Malaysian clinical trial in phase 1. And here's like I presented like 5 of the study that ongoing clinical trial in phase 1. Firstly, like we can see like NCT numbers, the ID numbers of each study that, as Dr. Tai he mentioned it about, like each of the study have to be submitted and approved by the FDA agency. And then the study title, you have to have your study titles, and there's we have 5 study titles in different condition, and then study URL, and then the condition I choose it 5 different condition, like as Yin she mentioned it's gonna be maybe in phase 1 only healthy volunteer, or the a specific condition like cancer, rabies like vaccine-based one, COVID-19, and iron deficiency anemia like metabolism syndrome, and also like each of the intervention conducted on drugs and biological things. So all the like the study that I have been like we choose it ongoing clinical trials, they are working on the drugs and biological intervention, and starting date of each of them like all started, most of them like starting in 2023, so it means like they will finish in the 2024 next year, because they have in the first one we have short period. So in the second slide, next slide, like I'm talking about the phase 1, one of the example of the phase 1 clinical trial that conducted in China's study ID, it's NTQ1062 in China's patient with advanced solid tumors. So the condition of the patient that they have been chosen for this study, all of them in advanced solid tumors. As we can see, like this study is open label and single arm phase 1 study that evaluates the safety, tolerability, and pharmacokinetic, and also preliminary pharmacodynamics effect of this drug in patient with the advanced solid tumor. So it means open labeled, that it means the patient know how only they know like what they will get the drugs. And the secondly, it's single arm. Single arm, it means like there is only one groups of the patient that undergone the study, and the number of the patient also the phase 1, in the phase 1 they conducted this study for the dose escalation to find out about the maximum tolerated dose of that drug on the patient. A second purpose of this phase 1, the dose expansion. The dose expansion is all about pharmacodynamics effects, and also tolerability of the maximum tolerated disease, and also finding the dose for second phase. Phase 2 trial, and the number of the patient, it was 32. It was small numbers of the patient, but it's phase 1, it's acceptable. And also starting date they started in 2021, and they will finish in 2024. As Yin she mentioned it, we have dose escalation. In dose escalation, so it means there is expansion in the time of conducting the study. Yes, how the study is designed. The important thing about the study that we are studying, we are like, reading about it, it's study design. First, this study is about treatment, as I mentioned, and then there's no allocation because single arm and interventional model, a single group assignment, and there's no any masking. All the patient, they know what they will get as an intervention. So in the study, we have to look for the primary outcome, what they are looking for it. The primary outcome, it's about maximum tolerated dose, recommended phase 2 dose, and also adverse events. If there is any adverse event, they have to stop this study. The secondary outcome about this study, it's about like pharmacokinetic parameter, objective response rate, and disease control rate, also duration of the response, and progression-free survival of the secondary outcome. So it's all about the mechanism of the drug inside the human's body. Here's like here you can see 5 other completed studies in phase 1. Some of them they pass it, and some of them they cancel it, but most of them they accept. As I named them, or I founded them in under the category of completed one, so they finished one, but most of them that I couldn't find the result of them. So, and there's also the same thing, like there's the ID, and also when they finish it, when they started and when they finish it. So the second, thank you. So next slide, it's about the completed study. So in the completed study, what we are looking for it, how we are interpreting the completed study. This study is about safety and immunogenicity of SARS-CoV-19 messenger RNA vaccines. It's about biological drugs, biological thing. So in China, it's the same thing. There's ID of the clinical trial, and what's about the study overview. I'm not going for the study overview. I will just talk about the result of this study, because because here's we need to look for the result. So in the result, firstly, the study contributed to our the result. There is no serious adverse event, so it means they pass it from phase 1 to phase 2 because there is no serious adverse event observed. And also both younger and older participants during the 6 months follow-up period. The result, the second one had antibody titers. The second one, titers were observed in younger participants. They found out that the antibody rises in younger participants increased more than in older aged one, and 30 microgram dose induced higher level of antibody, and this dose also declined more slowly by 6 months. So they knew about how immunogenicity worked in this vaccine. At the end, the study contributed to our understanding of the BioNTech pipeline of vaccine development and the safety of this vaccine that's conducted by the BioNTech company. Thank you so much. This is all about our clinical trial in phase 1.
All right, thank you very much. So that was a very informative presentation. I think you covered all of the bases, even more at certain things. So I'm very happy with the presentation. I hope everybody was able to follow. Now, Q&A time. Anybody got any questions to ask Ian and also Sana? Any question?
Yes, please. Any question?
If nobody has questions, maybe I should set questions based on their presentation. No question? Okay, if there's no question, I would like to ask. So, in the early part of your presentation, you mentioned about PK, PD, and what's the other one more term? Pharmacokinetics,
Safety.
pharmacodynamics, and no. There's 3. You might want to show back your presentation.
Yeah, hold on. This one. This one.
Yep, just pass already. The 4th slide. Toxicokinetic. Okay, so what do you understand about the difference of these 3 things?
Okay. I wrote here something. Pharmacokinetic is the study of what the body does to the drug. Pharmacodynamic is the study of what the drug does to the body. And toxicokinetic study is something like, I'm not sure, but is it the levels of drug that will cause toxic to the body? Something like that.
Yes. It's about the
So,
dose
Yeah.
of toxicity.
Yeah.
yes. So pharmacodynamics basically tells you what the drug does to the body in terms of whether it actually causes some sort of changes or not. Okay. While pharmacokinetic means that how fast will your body actually either destroy the drug or absorb the drug. So what the body does to the drug. Okay. Now, after you know what the body does to the drug and what the drug does to the body, you will also want to know whether it's going to be toxic to the body or not. So that is why these 3 key things are important. Okay. And when these 3 key things are known in an animal model, okay, one can then use, you mentioned something about using the body surface of the person, right?
Yes.
Yeah, okay. So I'm not too sure about using the body surface, but most of the time people would basically use the weight of the drug to the weight, so the drug to body weight ratio. Okay. So this one you put body surface area. This I'm not too sure, but normally it's through body weight. So for example, if I say the mice tolerable level is 1 nanogram per gram of the mouse is tolerable. So then when you want to scale it up to a human, say for example, the person is 50 kilos, okay? So then you want to then convert that number to the kilos, all right? So that is why sometimes when we look at results from researchers in papers and everything, it becomes very uh, uh, dumbfounding, because for example they say you need to the mouse can be treated with 1 milligram of the drug. So imagine if a mouse which weighs about less than 100 grams, you need to give 1 milligram. Then for a human which is, say for example, 50 kilos, that person will be 500 times more. So which means you might need to go up to 500 milligrams. Okay, 500 milligrams still possible. But if let's say it's 1 gram and then you need to give 500 gram, then it doesn't make sense anymore. Okay, so that is why it is actually crucial for people to understand this. Okay. Now, there's also one part that I want to ask you all. Can you please go to the part where you're talking about the different dosing, ascending dose, and all those things? The 3 strategies that is used.
This one? Dose
So
increment?
the
Of
one
this?
that you have. No, the one on number 15 and 14.
This one.
Okay.
Study design.
So yeah. Okay, so in this part, I don't really have a question, but it's more towards a comment so that your colleagues would understand. When people look at drugs, certain drugs have longer half-life, which is crucial. Some drugs you don't want too much of a half-life. Now, if I were to pose this question to everybody, what sort of drugs would require a longer half-time, while what sort of drugs would require a shorter half-life? Anybody want to answer?
Sorry, doctor, can you just come up with the question again?
Okay, so if you notice here, there is dose increase, and then you can see that some of this is, for example, like the study design in this particular slide. So the first cohort is given a dose, and then the second cohort is given another dose. The third cohort is given another dose. Okay, and then when you move to the next slide, okay, this one is given similarly. Okay, not similarly. That means in one cohort it's given in a dose escalation manner, and then the last one, okay, this is given alternatingly. Okay, so this is how they do it, correct? But
Yeah.
when is it that it is important to have low half-life drugs, and when is it important to have high half-life drugs? Which means which drug you want more time to be in your body, while what other drugs you would want it to be as little time as possible in the body. Can follow my question, or totally cannot follow?
I don't know, kind of like, but I think like biological drugs, mostly like it's need like low doses until get to the, the dose it will be tolerated or be accepted by the body without any side effects.
This is not okay. So tolerance is a key word there. Okay, so maybe just let me just put it in this way. What drugs you want to last a long time in the body, and what drugs you don't want to last a long time in the body?
Okay. What about the size? I think the size of the drugs also like.
Well, the size of the drugs determine whether it stays in a longer time or not. But what type of drugs you want it to be long lasting in the body, and what type of drugs you don't want it to be long lasting in the body?
I think it depends on what are the effects that you want and what are the disease that you are treating.
You are getting somewhere.
So if you are treating something like you want this drug to have a very long lasting effect, then the PD of the drug will be longer. And then if you just want this drug to work something like very transiently, then it will be very fast. I mean, something like that.
Okay. Now, what is the two? Okay, when we talk about diseases, what are the two main word that we use to represent a disease.
I was thinking about chronic and acute, doctor.
Correct. Very good. Okay.
Yeah,
So,
just now your question is the drugs typically you want to stay longer in the body is that the drug that can treat the chronic disease, so I guess.
correct. Very
Yeah.
good.
If a shorter one is a vice versa lah. Correct or not? I'm not sure.
Correct. So if you are
This is my
treating,
bold guess.
correct, it is a very well calculated guess, and it's actually correct. So you want, when you're talking about drugs, if you're treating a chronic disease, of course you want the drugs to stay as long as possible in the body so that it has more time to treat the disease. Okay, but if it's a disease that is acute, okay, it comes up very quickly, it goes away very quickly. You also want a drug that don't stay too long in the body. All right. Now that is only one criteria. There's another criteria that determines whether should a drug or can a drug stay longer in the body, or should or can a drug stay lesser time in the body. What criteria do you think that is?
The
Is it the
tolerability.
toxicity?
Yeah,
Yes.
the tolerability of your body towards the drug.
Okay, so it's the toxicity as well as the tolerability lah. All right. So if you notice that when you look at drugs, there's always side effects. Okay, be it nausea, be it gastric, be it whatever. All right. So the thing with drugs is that if, let's say, a certain drug, even if you need to treat, for example, chemotherapy, for you to treat a chronic cancer, all right, you don't want the drug to stay too long in the body as well. Why? Because the drug is cytotoxic. All right, which means it not only kills the target cells or the cancer cells, it could also kill other bystander innocent cells. Okay, so those are the situations whereby you don't want the drugs to stay too long in the body. All right, so the toxicity matters. So that is where you then come back to, at the end of the day, you need to look at pharmacokinetics, pharmacodynamics, and toxicokinetics. All right. So that is how you determine what and what during that sort of period of time. And that is also what actually determines your study design consideration. Okay? Everybody understand?
Yes.
Yep.
Okay. So generally, phase one, sometimes we use healthy patients, like for example, in the COVID vaccine vaccination studies. So that one, they will most likely use healthy patients. But if you're trying to come up with a drug that treats certain cancer, so that is when they will also recruit cancer patients. Okay. So okay, that is all from me. Any questions from anybody else? Going once, going twice. Okay, well done. Thank you very much, Yen and also Sana. So that was a very nice presentation. So please help submit the presentation into the website. Okay. All right. Now next. Give me a second. What happened? Oh, who is the next really? Give me a second. Let me just double check. So the next part is Jin Tao, Ziyu, and also Tapa. You will be doing phase 2 clinical trial. So please go ahead. Okay, who would like to start?
Okay. Okay, thank you. We will talk about
Go ahead.
the. Hello.
Yes, yes.
We will
Go
talk
ahead.
about the clinical trial phase 2. The number of participants in phase 2 clinical trials is moderate, about tens of people, sometimes over 100. They usually have one or two cancer types, sometimes more. The main purpose of clinical phase 2 trials is checking the best dose of treatment. Finding out more about side effects and looking at how well the treatment works. Sometimes it is randomized. Not all treatments tested in a phase 1 trial make it to a phase 2 trial, and about 33% of drugs move to the next phase. Phase 2 trials aim to find out effective new treatments that can be tested in phase 3 trials. To find out which types of cancer the treatment works for, more about side effects and how to manage them, more about the best dose. Phase 2 trials are usually larger than phase 1. There may be up to 100 also so people taking part. The length of study is several 2 months or 2 years. Randomized trials have at least 2 different treatment groups. Often there is one group who has the standard treatment they would have if they were not in the trial. They are the control group. People in the other group or groups have a new treatment or a procedure that is being tested. Sometimes there is no standard treatment available for the control group. This could be because people have already had all the treatments correctly available. In this situation, the people in the control group would have a dummy treatment. This is called a placebo. About placebo, some trials compare a new treatment with the placebo. It looks exactly like the actual treatment. It's the same shape or color of pill or size of injection. Comparing a group of people taking a new treatment with a group taking a placebo and can show if the new treatment is really working. A drug that actually works will work better than a dummy drug. About placebo, white trials. This is about placebo. White trials are randomized because researchers are possible to be biased without realizing it. For example, doctors may avoid putting patients who are more unwell into a particular trial group without real mean to. This leads to experimental error. The research team put information about the people taking part into the computer program. This might include the following: age, gender, the size of cancer, and how far it has spread. This makes sure that the groups are as similar as possible. Then the researchers know that if one group does better than the other, it's because of the different treatment and not because of general difference in the people taking part. Thank you.
Okay, so who is next on the presentation for phase 2?
Okay, so next is the pushback.
Hello, good afternoon, everyone.
Afternoon.
Am I audible?
Yes, I can hear you loud and clear.
So this is an example of ongoing trial, which is in phase 2. The title is Diagnostic Accuracy Study for OWL is the sponsored company and EVO1 is their product for a lung cancer. The study started in 2023, 10/5, and is estimated to be completed in 2024. For the enrollments, they are planning to take 350, out of which 150 cases will be the lung cancer patients and 200 as a control. It is an interventional study type. And there's no masking. Both the control and the cases will get or will have intravenously, the probe will be intravenously administered. And in the phase 1, they demonstrated the safety and tolerability. So in phase 2, they aim to assess the accuracy of this probe. That is their primary objective. And then their secondary objective is to define a test protocol for breath sampling and to understand, again, safety and refine tolerability, and also to minimize the healthcare worker efforts for, so once they administer the probe, then they will collect multiple samples for up to 180 minutes post probe, and then do the test. So the inclusion criteria are aged 45 to 85, who are able to provide informed consents, and then who are receiving a CT, including the chronic thoracic lesion with the last 6 months. Thank you.
Okay, next.
The next part is me. The example 2 is about the complete clinical trials to phase 2. I choose a simple example about three different liquids for COVID-19 patients. They aim to assess the ability of regular gallon to emulate COVID-19 in the throat and nasopharynx in stage 1 COVID-19 patients. So in the complete example, we should be include the clinical trial identifiers, the time of the of the post and results and sponsor. And about the study details, they divide the 4 groups. The Batavian, one of the Listerine and tap water, and the last group is no intervention, is the control group, and they should choose who will join these clinical trials, inclusion and exclusion. Next is the outcome measurement. Usually we have the primary outcome measurement and some secondary outcome measurement. And you can see that they will have the different time points. And next is about the study results. It may be include the primary outcome and secondary outcome. And the most important is the advanced events, the all-cause mortality and several advanced events and other advanced events, no including the serious. And this example is about the HPV vaccines in different clinical trial phases. You can see in the phase 2, they are more focused on the effect of the drugs, something like the CD4 cells and HIV virus loader and the cell-mediated immune response, like B cell response and T cell response. And you can see there are so many different between phase 1 and phase 2. You can see usually we will choose more patients. We'll choose more patients in the phase 2. But in this case, we choose more participants in the phase 1. It's about 961. But in the phase 2, they only choose the 150. And loss end is different because in the phase 1 they choose the healthy woman, and the phase 2 they choose the HIV infected females. And they are different about the time point. Usually we will say the during time in phase 1 is shorter than phase 2, but in this case, you can see the number of series converted subject antigen HPV6 and N18 amounts until to the 48 during the safety follow-up phases. But in the phase 2, they are shorter than 12 months. Okay, that's all.
Okay, thank you very much. So, any questions from your friends? Okay, so Yen asked, let me see what's the question. What is the primary and secondary results? What are the difference between these two results? Anybody want to answer? So if you see in the chat, Ian asked, what is primary and secondary result, and what are the difference between the primary and the secondary results?
In my view, I think something like in this case, in the phase 1, there are three there are primary and secondary the results, and in the phase 2, they only have the primary results. So in the phase 1, maybe they are focused in the safety, and in the phase 2, they may be more care about the effective about the drugs, I think.
What about the rest of the team? Do you think it's the right answer? Or do you all agree or disagree to that? Papa and Sri?
Yeah, agree.
What means the two type of
Sorry, Jintao?
you can say what means the two types of results?
Yeah, it's the two types of the results. Okay, does all phase 2 come with two types of results? If you notice, Ian, basically phase 1 has got primary and secondary results, while the phase 2 here in this particular study has only got a primary result.
No, I'm asking. Actually, I don't know what is the difference between the primary and secondary result. Why is it primary and why is it secondary? Why are they categorized in this kind of format?
Okay, I think I should step in here, lah. So, if you look at the phase 1 study, right, what is the purpose of the phase 1 study? It's for you to understand the safety and the dosing of the drug, correct? Correct or
Yeah,
not?
yeah.
Right? So that is actually or normally your primary results. Now, if let's say in the phase 1 study, you conduct it on patients as well, okay, what are you going to get? You're going to get some sort of information, for example, to see whether if there is a possibility of finding efficacy. Okay, so if you look at the primary results here, it's written as titers of anti-papilloma virus 16 and anti-papilloma virus 18 antibodies. Okay, number of subject with report of any or grade 3 solicited. Don't change. Go back.
Oh, okay.
Go back to the previous data. Okay, so number of subjects with any grade 3 and related solicitor general symptoms. Okay, so if you notice, right, number one is basically telling you what sort of antibody titer you're generating. Okay, so that one in a sense is really efficacy. Okay, and then the number 2 and number 3 is basically safety, to see whether there is any solicited local or general symptoms, which means that there is any adverse effects or not. Okay. Now, then, if you look at the secondary result, okay, so number of subjects reporting clinical relevant abnormalities in biochemical and hematological laboratory parameters being assessed. So this is not directly related to the things that you want to understand. So this is like a cascade of a cascade. Okay. And then if you look at the second part, number of seroconverted subjects against HPV 16, HPV 18 at months 12, 18, 24, 36, and 48 during the safety follow-up phase. So this one, in a sense, tells you whether the subject has got seroconverted or not. And then you look at the next part, subjects with pregnancy outcome. Okay. The second last one is related unsolicited adverse events. Okay, and then onset of chronic disease, serious adverse events, autoimmune disease, medically significant conditions. So now, if you look at the primary, is what you want to know, which is directly related to the vaccination. Okay, but when you go into secondary, it's sort of other things that we think might happen, okay, but we are not sure. So for sure, we will know whether we're going to get antibody titer, and for sure, we'll know whether there is a direct adverse effect or not. Okay, but then this one moves on to the secondary. You see, even pregnancy was being taken consideration too. Why? Because when you're giving a HPV vaccine, okay, they don't know whether the HPV vaccine will actually cause people to lose out on pregnancy or not. Okay, so maybe they want to follow to see that, okay, after vaccination, will it actually cause people to be unable to give birth? So that's why they have to look at pregnancy. Okay, Then they want to see whether there's any seroconverted subjects or not at other months to make sure that there is possibility of longer lasting effects. Okay, so all of these are actually very based on how it's being justified. So primary is always why I want to do this research or this clinical trial, and the secondary is always what might happen if, let's say, when we sit down and we think a little bit further, what can we actually see? So it's like sometimes it's something which is the researcher or the PI, the principal investigator, didn't actually thought about it in the first place, and then after that it was reported, or after the initial part that they do this, then they think, okay, maybe this part we can also look into, but this is not as critical as primary. So that's why there is primary and secondary result. If you look at clinicaltrial.org, you will always find primary and secondary result. Okay? Understand? Jin
Okay,
Tao,
okay.
Papa, and also Ziyu. Yeah. Okay. Any other questions from the group? Okay, I would just like to comment a little bit more on phase 2 clinical trial. This one maybe you guys missed out, or I'll just try to sort of summarize it a little bit. So phase 1 always looks at safety and dosing. Okay, when you go into phase 2, it will still deal with safety and dosing. Okay, you will still need to see whether there is a certain level of safety to it or not. All right, but at phase 2, you want to start to see efficacy. Okay, because at the end of the day, you still need safety to come in first before you look at efficacy. It's no point if treatment or a therapy is efficacious, but it's not safe. Okay, but there are also sometimes when even if there are safety issues, there's always a balance. Okay, for example, treatment A would help 100. Uh, okay, treatment A will help 95% of cancer patients. Okay, but treatment A also might cause serious problem for 9 or 10 patients out of the 100 patients. Okay, say for example 10 to 15% of the patients. So if that is the situation, people might still consider, and then they will also think, okay, if it protects or if it helps 95% of the patient, these 10% of patients that have negative effect, is it something which is tolerable or is it something that would lead to death? Okay, if it's something that would lead to death, then totally it's out. Okay, but if there is like a certain level of let's not go into disability lah. Something like, for example, they might have a certain problem with their nervous system, or they might have a certain central nervous breakdown, or maybe a week or 2 weeks. So those are still considerable as tolerable events, adverse events. But when you come to serious adverse events, autoimmune diseases, look at the secondary results there. Autoimmune diseases, new onset of chronic diseases. So these are things that people would actually avoid. Okay? So no matter how good your treatment is, they will still avoid. All right. But if let's say it is the treatment actually is available for certain side effect, they might actually go on with it. Okay. So number one, safety for phase 1. At phase 2, safety is still there, but you want to start to look at efficacy already. Okay. All right. So that's my comment. Any questions? If no questions, we'll proceed to the next group. Nana. Okay. So the next group will be Rad and Zian. You'll be doing phase 3.
Okay. So, oh yeah, by the way, my name is Zhijian, not Ziyan.
Zian. Oh, you sorry lah. Must be typo.
Hold
Never mind,
on.
just call it video
Let
lah.
me check for my this.
Never mind, I'll just go
See my
I'll
slide.
just go with video lah. Easy.
Can you see my slide?
Zian. Oh, okay. Yeah.
Can you see my slide, everyone?
And
Wait, I can't hear you. Is that my problem right now?
you cannot hear us. So
Hold on. Okay. Can anyone say something?
Hello.
Oh yeah, good. Okay. Was worried for a while. So yes, all right. So welcome to my presentations. So me and Rad together will present you about clinical trial phase 3. We are not going to into a very deep. We're not going to touch into a very deep point, but hopefully this cover a lot. This cover enough information. Okay. So yes. Clinical trial phase 3. So, clinical trials, by definition, is a research study in which one or more human subjects are prospectively assigned to one or more intervention to evaluate the effect of those interventions on health-related biomedical or behavioral outcomes. Simply means that this is a study research that to test your result in multiple subjects, and also you might have multiple drugs. This is the general definitions of clinical trials. So what's the difference between phase 2 and phase 3 is that the phase 1, phase 2, and phase 3 is that in the phase 3 is the aim is to demonstrate the efficacy and monitoring of the adverse reactions. So you can imagine it's like actually simulating the real world situation where you really prescribe this to someone and you want to see what happens to them in a longer term, right? So this is the main difference between phase 3 and phase 1 and phase 2. Okay, so let me move on. Next slide. All right, phase 3. So the study participant usually will involve to 300 to 3,000 volunteers who have the disease or condition. So now we are not talking about people with we're not talking about healthy subjects. We are talking about this patient with the related conditions, and the study duration usually will involve to 1 or 4 years or more. Okay, it depends on the design of your studies, but typically from the FDA definition they give is 1 to 4 years, but actually a lot of them is not only stop at the 4 years. Okay, 4 years is actually more than that. So okay, description about this. Researcher design phase 3 study to demonstrate whether or not a product offers a treatment benefit to a specific populations. So sometimes known as a pivotal studies because once this phase 3 is passed, actually more or less your drugs is going to be finalized into a FDA list already lah. So it's sometimes known as a pivotal studies. These studies involve 300 to 3,000 participants, and the participant and researcher are blind to the treatment group, so-called as a double-blind design. What does it mean is that the participants and researcher didn't even know, they don't know that they're being treated to this. Only the monitor knows. The participant and the researchers doesn't know about that. So phase 3 study provide most of the safety data. For example, it's possible that less common side effect might have gone undetected in the previous phase because previous phase wasn't taken too long. The time that you observe the condition of the patient wasn't taken too long. So so phase 3 is the time that we test whether your drugs have real adverse effect or not. Because yes. So however, as phase 3 studies are larger and longer in duration, the results are more likely to show a longer term or rare side effects. That's what I mean from there. So this is one of the few examples of the studies that ongoing in phase 3 in Malaysia. So we can see that this is AZDEX D505 versus placebo in men with metastatic prostate cancer. And then the other one will be how zitifekimab works compared to placebo in people with a heart attack. So actually you're actually comparing, like you're giving a placebo and also the real drugs and see whether what's the effect from there. Okay. So this is the definition of phase 3 clinical trials from FDA. Okay. So one of the things I want to highlight is that approximately only 25 to 30% of drugs move to the next phase, and this has has to do with some reasons that we're going to be discussed by Rad later. So yes, so want to take a look into this data. This is the data of oncology and non-oncology success rate by phase. So as you can see that to phase 2 and phase 3, the success rate is typically low compared to those to phase 3 from moving on from phase 3 to NDA BLA. So it means that most of the not most of the a lot of the drugs is still at the phase of the phase 3. So yes, so I'll pass the stage to Rad. So he will be continue presenting.
Okay. Next slide. Okay. Okay now?
Okay.
Okay. Hello everyone. Now I talk some problem with the drug made in phase 3. Made the drug depend two point, the safety or effect. Now why most of the drug fail in phase 3? Effect challenges in the demonstration of the drug effective, the drug okay, just a minute. No. Effective the drug. Safety issue, discovery in safety a problem according with the drug. A limited edit, preference, failure the active the significant the befend complement access the treatment. Okay. The effective study design, the failure study, the failure study, the study design, it can combine the study ability effective. In fact, patient solution, the conject patient effective result study ability, the drug prevent. Available in response substance viable in response a drug contact failure. Okay, Leo. Okay, sorry, found this other slide. Leo, can you hear me? Leo.
Right. I think you are presenting.
Leo, sharing other slide, Leo.
Oh, you want Leo to share
Some
the other
problem on
slide?
internet.
Leo, can you please share the other slide?
Okay, now, okay. The driver of the failure. Okay, now, seen?
Yep, can you see?
Can you see now?
Yeah, we can see.
Okay. Depend basic the science. This found the some example basic science the beneful effective animals method reproduct poor useful for the target disease biology. Flow the study design. The sustainable does the science flow the data collection analysis a problem with a study option. This some example about of this problem. Okay. Okay, thank you.
Okay. Thank you very much, Rod and also Leo. So, any question? Any question from anybody? Nobody? Okay, let me ask a question. So in the clinical trial phase 1 and phase 2, normally it's being done at a single center. Okay, so for example, if it's done in hospital, Penang General Hospital, so that is where it's being done. What about phase 3 clinical trial? Is it done at only one location, or is it done in multiple locations, or how does it go?
Yeah, I forgot to mention this. Actually, phase 3 was done in multiple sites. So as you can see from the example, where's my mouse? Oh, wait. Why,
You're
why
not
so?
presenting, Rod is presenting,
Okay.
Oh, representing.
Okay, Leo now?
Wait, the WebEx is lagging.
Especially when you open too many things, it will lag.
Like this. So, yes. Oh, it doesn't mention here. Okay, good. No. Typically, I want to say is that phase three clinical trials is happen in multiple site. What does it mean by multiple site? It's not just only few hospital in the regions. It's actually involve, let's say today, this drug is being is coming out from a pharmaceutical company like AZ or Novartis. Probably the tested site hospital will be involved in multiple countries, a lot of countries. So if you like, I can actually share the internet result lah. Hold on.
It's okay, you don't need to share, you only need to explain.
Okay. Yeah. Okay. I know it. Share. Okay. So yeah.
Okay.
So actually, if you see from here, right? Let's say today this AZD5 versus placebo in men with metastatics, castration-sensitive prostate. As you can see, the location is 334 more locations, which suggests you that clinical trial phase 3 is happening in a lot of different countries, different locations,
Okay.
a lot of different locations.
Okay, so now my next question: why does it need to be done in multiple locations?
Because you want to get as randomized data as possible. I didn't read about this, but if I want to take if I want to come up with a good reason about this is because the genetic population genetic is diverse according globally. So you might have these drugs work in for the, let's say, for the Caucasian or for the Angmo or the white people only, and not for the Malaysian like us. So that's why you want to have to try in different people or places.
Correct. So one of the reasons why multiple centers is required is because you want to make sure that your drug is not only beneficial for one genetic population. All right. It has to be suitable, and also it has to be efficacious for people in various populations. Okay, so that's why you need to do a multi-center. Don't forget, your phase 3 is for you to further determine the efficacy as well as to find any other possible side effects. All right, so that's why you have to put it in multiple populations. Say, for example, the genetic population of people in the US might be different than from the one in Malaysia. Okay, maybe because of our lifestyle, maybe it's because of the genetic pool. So all of these might lead to different this one. There's also things like, for example, if you're talking about pharmacokinetics and everything, if you're going to look up cytochrome P450, okay, that is an enzyme that sort of a measurement of an enzyme that actually looks into how well your body metabolizes certain drugs and how bad your body metabolizes certain drugs. Okay, so because of that, you need to basically test it in multiple population, as many sites as possible, so that you will be able to have a drug which is which you can understand. Because at the end of the day, pharmaceutical companies will want to sell the drugs worldwide. All right, and a drug that is being generated must also sort of be efficacious, not efficacious, must also be contributing to the global population. Okay, so that is why you need to do multi-site evaluation, and that multi-site evaluation will basically give you an idea of whether it is efficacious for certain pool of people, not efficacious for certain pool of people, or it could have side effects which might be terrible for certain people and not for certain people. Okay, so that is why clinical trial phase 3 is so big and will require multi-center. Understand?
Yep.
All right. So, any question from the rest? Any other questions? No? Okay. If none, then let's go to the last group. So clinical trial phase 4. GIE and Sultana, you all go ahead.
Okay. Our group's topic is phase 4 clinical trials, and the team member includes Tana and me. Clinical trial phase 4 also known as post-marketing research refers to research on the real situation and long-term safety of drug use conducted after the drug is listed. Phase 4 trial is a supplement and culmination of phase 1, 2, 3 trials. In addition, compared with the clinical trials in the first 3 stages, the phase 4 trials pay more attention to the application and the effect of drug in real clinical practice, which is more realistic. And the research purpose is to investigate the efficiency and the adverse reactions of drug under a wide range of use conditions, evaluate the risk and the benefit relationship under the use in the general population or special population, adjust the dosage and discover new indications. And the mean population recruited is a broader and diverse group of general patients or special populations. Generally, the sample size of subjects is greater than 2,000. There's no need to set up a control group, but it also does not rule out small sample randomized control studies for some indications or some research objects as needed. Mortality and mobility outcomes are often used as one of study endpoints. And the reason why we need to conduct in phase 4, because phase 4 clinical trials are more comprehensive than previous trials. Other trials may be limited because patients need to follow strictly prescribed dietary and drug regimens, which phase 4 trials are conducted in the general population to investigate the effects of various food and drug on the drug performance. Such a design can more comprehensively evaluate the real effect and safety of drug. The phase 4 trial was conducted in multiple centers and different populations, eliminating potential bias and off-field effects and increase the reliability of study results. In addition, phase 4 trials help to find real adverse reactions that may be missed in other trials and improve the comprehensive understanding of drug safety. Some drugs were exposed to harm effects in phase 4 trials, leading to their withdrawal from the market. And therefore, phase 4 trials are essential to ensure drug safety and provide comprehensive data for regulators to evaluate the performance of drug in the global population. And the next part goes to Tana.
Yeah. So these are the examples for the phase 4 clinical trials. So so basically, phase 4 clinical trials are done for knowing the longer adverse effects of the drugs in a larger population. So example 1, this is the title of the example. Yeah, indication, it's done for the efficacy, like the combination therapy and one monotherapy. They are like um, comparative efficacy of the combination. So the study started at 2024. Study will get completed in 29. So it's an ongoing trial, and enrollment is 6,000 plus. Study type is interventional. It's a phase 4 trial. Population: patients having coronary artery disease. Ages eligible for study are 19 years or older. Sexes eligible for study are all except healthy volunteers? No, because this study is um the patients involved for this study are coronary artery disease. So yeah, example 2. Yeah. So the next example is for the completed trial, phase 4. So it's the title. And for this trial, the indication was adverse events. So they wanted to know the long-term effects of the vaccines. after effects of the vaccine. So enrollment was 580. Study type is interventional, phase 4 in healthy population. Yeah, they accept healthy volunteers because this is a vaccine trial. So yeah, those are the examples. And thank you.
Okay, thank you very much. Any questions? Okay. Why is it that some drugs need a phase 3 phase 4 clinical trial while some drugs doesn't need a phase 4 clinical trial? Anybody? Anna, PIE?
Yeah, here.
About like monitoring the drugs after being approved in the clinical trials in the Finland.
Why is Anna answering? I'm asking the
Like
people
my
who are
graphic,
presenting.
sorry, like it's not working very well. I cannot even hear your question very well. That's why I cannot even answer.
Okay. No, I'm asking why is it that certain clinical trial, why is it that certain drugs need to undergo a phase 4 clinical trial while others don't need to go through a phase 4?
Because of the patient population is small, so they will not go to the phase 4.
Could be as well, but there's another more compelling reason actually. Anybody? Okay.
Is it because all the information is already established with all the phases? So maybe there's no need for phase 4?
Not
Like the
really as
safety
well,
is.
because if you notice, phase 4 is also out there to look for possible possible side effects, right?
Yeah. I think this is coming from the genetic engineering drugs, the one that basis of genetic engineering nowadays, like most of the drugs being approved, like one of them like HGV being approved, okay, being like used in patient, but in the case it has to be under the evaluation and monitoring till 10 years after being approved because of there is no enough data about the uh, how that genetic tools will affect the humans inside the human cells because they conduct it in the ex vivo, like outside of the cell, they change it outside of the human body, they change it the cells, genetic engineering, and then they increase it back to the body cells. So there is no like evidence like what it will happen inside the body. But they figured out will happen inside the cells. But inside the human cells, there is no any data. So they like just extend this period to be monitored and under the uh, control of the finding out about other like side effects of this drug.
Well, that is also very possible. But the thing that I was looking for is actually, if you notice just now when we were talking about phase 3 clinical trial, right? We look at the one that Leo shared, it was about 384 locations, all right? But the thing is that could they actually cover all the continents or cover all the hospitals or cover all the patients? It's not possible, right? So just now, I think when Tana mentioned about the word insufficient patient during clinical trial phase 3, that actually somehow plays into the reason. But the main reason is so that there are certain drugs which must be tested more widely because in a sense you only cover, for example, some clinical trials phase 3 takes part in the US and Europe. So at the end of the day, you're still covering Caucasian, right? But the drugs would require you to be you want to sell the drug to India, to China, to Southeast Asia, to Australia. So the population genetics might not be sufficient. So in order for the drug company to actually convince people to use their drug, they would then initiate the phase 4. Don't forget, when you do phase 4 clinical trials, the company or the drug company that's actually selling the drugs or is marketing the drugs would have to fork a certain amount of money out for it to be done. So it is going to be a very expensive process. So in order for them to, the only reason why they would invest in such an expensive process is when they know they want to sell it to more places. Okay, so at the end of the day, a phase 4 clinical trial is for two reasons. Okay, number one is marketing, all right, whereby the drug company wants to sell it to as many people as possible. Okay, and number two is so that the any sort of adverse effects which were not seen in certain population can also be discovered. Okay, yes, Ian?
When you talk about you want to market to as many population as available, means if I only study until clinical phase 3, I can only market to this country, but not other countries.
No, you can market to other countries, but who actually decides whether they will take your drug or won't take your drug?
Oh, okay.
You first
Their
need to
own
get buy-in
country
from.
regulatory lah. Is it?
Well, normally regulatory, if let's say if it's FDA approved, the regulatory people will just go through the data one more time, and if they find that it's okay, then they will agree to it. Okay, but the people who actually decide to sell your drug to the patient will be the doctors. Okay, and the people who is going to decide whether to take your drugs or not is going to be the patient. So the doctors and the patients will basically be the people that you need to convince. Okay, so when I say marketing, it's more towards have this particular drug been tested on people of my kind. Okay, if it has been tested on people of my kind, then I have a much better what's the word for it? A much better level of acceptance to it. Okay, so that's why I say phase 4 sometimes is more towards marketing, because you need to convince people to use your drug. For example, if drug A's efficacy is 70%, okay, it has been in the market, then suddenly you come up with drug B and you say your efficacy is 80%, 10% increase, okay, but it hasn't been tested. It's just like, why is it that there are people who didn't want to take the Pfizer or the AstraZeneca vaccine, and they prefer to wait for the Chinese vaccine, Sinovac? Because mRNA vaccine has not been tested before. All right. So you would want to stick to your Sinovac style of vaccine, okay, whereby it's basically attenuated virus. Okay. So at the end of the day, it's up to phase 4 is more towards convincing the doctors as well as the end users. Okay. However, if let's say you have a drug that you have a disease that you have totally no cure, okay, and this particular drug can cure the disease, okay, if you are a doctor, you would want to try to prescribe that drug to your patient, okay, and if you are a patient, you know that there is no cure except for this particular drug. Do you think you want to try it? I'm pretty sure you want to try it, right? But if let's say there's really a drug that has a 70% chance of curing you, and then these drugs come along and then they say, oh, you got an extra 10% chance. So then people will say, so what? It's not tested. I don't know whether it's safe or not. So at the end of the day, clinical trials is not only to convince the regulators, but it's also to convince the people who take it. And that's why I say sometimes it's more part of marketing rather than the rest. But of course, it also comes with scientific merit, whereby people know that in a certain population that hasn't been tested in the clinical trial phase 4, they might still be able to find a lot of underlying adverse effects or side effects in different type of population. And then basically it allows, or it basically adds value to the drug for the drug company. Don't forget, phase 1 all the way to phase 4 is all sponsored, which means that the patient who is undergoing it not only do not need to pay for the drug, sometimes they even get paid, okay, a certain level of compensation. And that's why just now you're talking about phase 1, whereby for clinical trials, you're not supposed to, undergo multiple clinical trial. And then sometimes if it's radioactive, radio labeled drugs or something like that, you need at least 1 year before you can do another trial. Why? Because clinical trials are paid for by the sponsors. Okay, for those people who work in or were a student before in a research institute that does a clinical trial, normally you get offers. Would you want to try to undergo this clinical trial? There is a honorarium or a compensation of 400 ringgit, 500 ringgit, depending on what is the level of monitoring that is required. If it's just taking blood after you take a certain medication, maybe they'll remunerate you with 100 ringgit, 200 ringgit. But if it's something that say, okay, you need to take this drug every 1 week, you need to come back to do a blood test, then that one might be a few hundred ringgit. Okay, it's the same in Malaysia. It's in the same in other countries as well. All right, so all of it is being paid for. And the only reason why the sponsor will pay for it is because they know they will recoup the amount if, let's say, they get buy-in from patients as well as doctors. So as long as doctors buy in the whole treatment, then there's a very likely chance that they will be able to recoup whatever investment that they have made. Okay, so at the end of the day, all these clinical trials, yes, it is good for humankind. It is important, especially for safety, as also for efficacy. By the end of the day, if let's say you have a drug which has got no clinical trial data versus a drug that has got clinical trial data, which one will you go with? You definitely go with the one with clinical trial data, right? Because everything, be it from safety to efficacy, will be okay. Okay, so that's why you would then take that drug compared to other drug which they have some wild claims, but then there is no any clinical trial before. Okay, because there are still markets out there that actually have drugs or sell drugs which are illegal. So having clinical trials is not only a balance between profit or assuring the patient, it is also a way to sort of counteract with possible dealings of illegal drugs and everything. Okay. All right. So I think with that, I'm just going to quickly summarize. Phase 1 is a very small trial just to look for safety and also dosing. Phase 2, you also look for safety, but you start to look into a little bit more into efficacy. Okay. But this two is just based on a smaller population at a smaller genetic pool. When you go to phase 3 clinical trial, you want to look for efficacy across the board, more towards international community, as well as to see whether there's any potential side effects. So safety comes in for clinical trial phase 1, phase 2, phase 3, even phase 4. Okay, because phase 4 is where you also find some underlying clinical, some underlying adverse effects or side effects as well. So if you notice, all 4 clinical trials will always have safety, and efficacy only comes in phase 2. Okay, so always look into safety first before efficacy. All right, so that is sort of the conclusion for all the clinical trials. And for you to actually be involved in clinical trials, it's not only just you doing something, it's normally part of a bigger group. And therefore, clinical trials, especially phase 2 and phase 3, are normally taken care by pharmaceutical companies, because that is where they have the money to do it, while people in the university just might not have sufficient money to do it. Okay? Understand? Cool. So, any other question?
Yeah, Doctor, I have one question.
Go ahead.
So, when I can sell the drugs after the 3 phases,
Mm-hmm.
I I mean, after the phase 3, I can sell my drugs,
Mhm.
or I need to finish all of the process if we need to do the 4 phase.
No, as long as you finish your clinical trial phase 3, it shows efficacy and it's safe, you can actually start selling the drugs.
Oh, yeah.
Right?
Because I
And
see a case,
normally-
they pass the 3 phase and then they sell the drugs and then they do the phase 4.
Yes.
Yeah.
So normally, after phase 3, you can start to sell your drug. Phase 4 is normally when your drug has been started to be sold in certain countries. Then you want to penetrate other countries, then you initiate it as a phase 4. Okay, say for example, the drug was created in China. Okay, it has been it passed clinical trials phase 3 in China, in India, in Japan, in Korea, in Malaysia. Okay, but it hasn't done any clinical trials in Europe or in US before. So if the company then decides to push the drug to Europe and also US, they will most likely sponsor a clinical trial phase 4 over in Europe as well as in the US. Okay, and then once they penetrate into those markets and it's efficacious, they are able to convince the users there, then they will be able to sell in those markets better. But at the end of the day, they also get other adverse effects data or other efficacious data. So maybe it works. It cures 90% of cancer patient in Asia region. But when it goes to Europe, it cures 95%. So then that becomes an additional selling point. But even if, let's say, it doesn't cure 95%, it maintains at 90%, it is still good because then there is sufficient data to say that it works for that particular population as well. All right.
Okay, thank you.
Okay, any other question? So, Ian, you were asking emergency need of drug, somehow come across the word orphan drug. So, emergency need of drug is normally like the COVID vaccine. So they showed in phase 1 and phase 2. To a certain extent, they show efficacy, and because of emergency, they had to just quickly push it out because there's already demand. Don't forget, it's always demand. So that time there was demand from a lot of countries. So that is why they push it into emergency. FDA has got no choice. They have to just bow down to the need of the world, and therefore they go for that. And in terms of orphan drug, is something that is used to treat rare diseases. Okay, so when there is a rare disease, okay, that has got no cure, okay, so a drug that is being researched for that could be used as an orphan drug. All right. So what is this YouTube link for? Yeah, what's this? The Intamus restaurant and shop weekend. What is this? Is this the CD28? Yep, cytokine storm. So this story, I know because it involves immunotherapy. So yeah, they were using an anti-CD28 that was tested in mouse. No, in monkeys. It works very well in monkey, but at the end of the day, they didn't realize that there was a difference between the human and the mouse and the monkeys activation markers. So your anti-CD28 basically antagonizes with CD28. So in the mouse, the CD28 expression is lower, but in human it's more. So when you over antagonize your CD28, what's going to happen is that what happened was that there was too much cytokines being produced by the body. So if I'm not mistaken, there were, I think, 3 or 4 patients who actually suffered from it. So some of them get amputated, and then some of them had got organ failures and everything. So yeah, that is quite a famous clinical trial phase 1 that actually failed. So that's why at the end of the day, safety is always the most important. Okay. All right. Any other thing? If there is nothing, then that's about it. Thank you very much, everybody, for taking time listening to my lectures, although sometimes might be a bit lengthy. I think this year's class has also been very interesting because you guys are a little bit more outspoken than the other previous groups. I like to have discussions instead of just me rambling around. So with that, thank you very much again for your attention and good luck for your finals. I also got all the the lab reports ready, and you guys will be able to get your results. I think Encik, Faiz or Bob will be able to share it with you all according to the time that is allocated. All right. So with that, thank you very much, everybody, for your time. That's all. See you around. Okay, thank you. Bye.